Hi everyone. So we're going to start getting into a little bit more depth on oncogenes and tumor suppression genes. So we're going to start out in 22.5 on oncogenes, and then we're going to talk about the genetic changes that will convert something that we would call a proto-oncogene into an oncogene. And then we're also going to talk about the different ways that these oncogenes can lead to cancer. There's some vocabulary too for this particular chapter. So when we think of proto-oncogenes, these are what I would consider a normal, non-mutated gene that has the potential to become an oncogene. And what I mean by that is that the particular gene can play a role in uncontrolled cell growth if it is mutated. So the normal non-mutated one is called a proto-oncogene, basically meaning that it's ready to become an oncogene, but there has not been the mutation to do that. Generally, what we see is a gain-of-function mutation that produces that oncogene. And what those effects could be usually fall into one of three categories. Either the amount of protein is increased when we get this gain-of-function mutation within a proto-oncogene. We could get a change in structure of the protein that makes it overly active so that cell division really is upped. Or the protein expression in a cell type where we don't normally see that particular protein expressed. And that protein then could go on to cause uncontrolled cell division. So keep in mind that when we think of the growth factor control of cell division, this is where we often see these oncogenes playing a role. So the cell cycle is a series of stages that the cell has to progress through that will lead up to eventually cell division. And there are certain places that when we go past it, that it has to occur. So we have the first gap phase, and this is when a cell may undergo molecular changes that will allow it to commit to division. So once it commits to division, then we go into the S phase, which is going to be DNA replication. We get the second gap phase, and finally that mitosis and cytokinesis, so cell division has occurred. So when we think about this, we can have commitment to cell division that is regulated through polypeptide hormones known as growth factors. As those growth factors increase, they then can bind to cell surface receptors, and they're the ones that will initiate these cellular events that will lead to cell division. And so as these cellular events occur, what we can see is that many of these cellular events will get changed in cancers, meaning that instead of having close regulation with these growth factors and cell division, we have an increase in those growth factors, and then that means that there's going to be an increase of cell division. And the one that's probably one of the most studied is this epidermal growth factor in skin cells. Epidermal growth factor is actually a growth hormone that tells a skin cell, hey, it's time to go through cell division. And you can see on this particular epidermal cell, we have these receptors that can be bound by the EGF. When that growth factor comes in, what it does is it sends off this cascade of reactions that tell the cell to go through cell division. And one of the ones I want you to really focus in on here is this RAS protein here. As GTP, which is kind of a subset of very much like an ATP, comes through, when it binds this RAS protein, what we see is the hydrolysis of that. It actually is what is going to cause cell division to occur. And in a regulated one, what we see is that this receptor will only have that growth factor when the cell needs to go through this. But what can happen is that with this RAS protein, that's a GTPase, so it's an enzyme that breaks down GTP, it actually is a proto-oncogene. And when it mutates, it means that it becomes an oncogene. What ends up happening is there's a missense mutation in these genes, and we see this in a large number of these RAS genes. And what ends up happening is those mutations then decrease the ability of the RAS protein to actually break down GTP, and it becomes bound by this GTP. And what that means is that the protein is in its active form, and it keeps that signaling pathway turned on, basically saying to the cell, you need to divide non-stop. And I have a quick picture of this. So what we normally would see is this GTP comes in, it activates our RAS protein, and then we get hydrolysis, and now we have GTP and inactivate this RAS protein. The cancer-causing mutation of this RAS protein means that it prevents hydrolysis, and we keep this GTP bound, and we keep telling the cell to go through cell division rather than having this hydrolysis. And so that is where we have gone from a proto-oncogene to an oncogene. So that's one of the ways, and in this case we saw it as a missense mutation, but there are 4 different mutations that we frequently see that can change a proto-oncogene into an oncogene. I just showed a missense mutation. We could also get gene amplification, which I'll show in a minute. We could get chromosomal translocation, meaning that part of a chromosome is translocated and it can upregulate that particular gene. And we can get viral integration, which can disrupt the proto-oncogene and turn it into an oncogene. So I'm going to go through each of these. The missense mutations that we just saw in the RAS gene can change that structure of the RAS protein and make it permanently activated. In the human genome, there's 4 of these RAS genes, so these enzymes that could be affected, and missense mutations in these genes are associated with different types of cancer. For example, a mutation in the RAS-H changes an amino acid in a protein from glycine to valine, which makes it an oncogene. Chemical carcinogens have also been shown to make these changes, to actually make these missense changes in the RAS genes. We can also get gene amplification of oncogenes, and so we can get amplification of a particular oncogene, which means that we have increased copies of the gene, which means more protein. And what we can see is we can get amplification of a particular gene here that can cause leukemia. Amplification of another one can cause neuroblastoma, and in this one it can cause breast cancer. So basically a normal amount of this particular gene and down the road protein does not cause cancer, but if we have an amplification, it means we have an overexpression of this gene. We can also get chromosomal translocation, which can form oncogenes. So some tumors will actually have specific chromosomal translocations. An example of this would be chronic myelogenous leukemia, or CML, and this is correlated to what is called a Philadelphia chromosome. That is actually chromosome 22 that gets shortened because it gets translocated to chromosome 9. This can result in this proto-oncogene being fused to a promoter and coding sequence, which upregulates then this particular gene. And I have a picture of that. So you can see we have chromosome 9 here and chromosome 22. We get translocation. This portion is translocated over to the 22, and what we get is a shortening of 22, but that translocation actually increases then the replication of BCR, and we get chronic myelogenous leukemia. The fourth way that we can see these oncogenes is viral integration. So some viruses can integrate into the host DNA and become part of their life cycle. These are what we call proviruses, and they can actually cause activation of a proto-oncogene using a viral promoter or enhancer. So when that virus integrates, it brings with it sometimes these promoters and enhancers, which will upregulate a particular proto-oncogene. And so a few of these viruses are known to cause cancer in plants and animals and humans. We call this transformation, where we convert a normal cell to a malignant cell. Most of these cancer-causing viruses are not very potent at inducing cancer, but there are a few types that can rapidly induce tumors in animals, and these are what we call acutely transforming viruses. These are ones that we find to be particularly dangerous. The one that probably was first discovered was the Rous sarcoma virus, or RSV, and this was isolated from chickens in 1911. The research of this has led to the discovery of many oncogenes and cancer-causing viruses. As a matter of fact, up to 15% of all cancers are caused by viruses, and I have a table kind of showing this. Some of the ones that we know have the real ability to do this are the retroviruses. These are RNA viruses that reverse transcriptase and then put their DNA reverse transcription into the host cells. We have ones that can cause sarcomas in chickens and cats. And then in DNA viruses that we know, especially dangerous in humans, are hepatitis B, SV40, and polyoma virus. These can cause liver cancers. We also have papillomavirus. The human papillomavirus is one that we know can cause cervical cancer in humans. And then the herpes virus. These ones can cause carcinoma in frogs and in chickens. The human herpes virus and Epstein-Barr virus can cause Burkitt lymphoma and occurs often in immunosuppressed individuals. So we do know that many of these viruses actually cause cancer and can make those oncogenes turn on. That's the end of 22.5, and we will be moving on after there's some problems here to look at, looking at proto-oncogenes and oncogenes. We will be moving on in our next lecture then to 22.6, which is tumor suppressor genes. So going back to this, you want to be able to describe the genetic changes that we see that can convert a proto-oncogene to an oncogene and give at least 3 examples. Also explain how the genetic changes are expected to alter the activity of that gene product. And then lastly, with regard to cancer cells, which of the following statements are considered true? This is just kind of a review. Hopefully this is helpful. Please let me know if you have any questions, and we'll be moving on to tumor suppressing genes in the next mini lecture. Thanks.